Hello my dear friends, I welcome you in my channel Eduscope. Today's video is about biotechnology part 2. In my last video, I discussed about the biotechnology, the introductory part, and I also told you that what is the importance of biotechnology and the main steps of recombinant DNA technology are selection of the desired character and isolation of the DNA having desired gene, fragmentation of DNA by enzyme, isolation of DNA by gel electrophoresis, amplification of DNA by PCR means polymerase chain reaction, ligation of the DNA into the vector, and the insertion of the recombinant DNA into the host, culture of the host in suitable medium, and extraction of the gene product. And last is downstream process, that is to obtain the finished product. And today I am going to discuss these three steps 1, 2 and 3 in detail, because it will take long time. That's why I divided this total process into different videos, and today's videos I am going to discuss this first, second and third. So first thing is tells about selection of desired character. What is that selection? First of all, we have to select which character we want, which product we want, and which gene is responsible for that product. That is the main thing, selection of characters, what characters we want, what product we want, that has to be selected. That is the number one. After that, second thing is very important, that is isolation of DNA. Second step is the isolation of DNA. Now question is, it is a very interesting process. Second steps is very interesting, as I told you, and these steps tells us about how to isolate the gene of interest from any organisms, how to extract or isolate the DNA having the desired gene. Okay, suppose this is the cell. Within this, it is a cell having membrane. Suppose it is cell wall. In case of plant cell, cell wall. Fungi, cell wall. And below of it, another membrane is present. That is present every cell, that is cell membrane. Cell membrane. Okay, and within this cell membrane or protoplasm nucleus is present, where this DNA is present, clear DNA is present. So we have to take out these chromosomes or DNA from the nucleus present over here. Now we have to break this one, cell wall from the plant cell. If it is plant cell, we have to break this one. After that, cell membrane. After that, nuclear membrane. Now we have to break one by one. First of all, cell membrane is broken down by cellulase, enzyme cellulase. So this cellulase enzyme breaks the cell wall because it is made up of cellulose. It is made up of cellulose. In case of fungal cell, it is broken down by another enzyme, chitinase. That is by chitinase. In case of fungal cell, in case of fungal cell wall is broken down by chitinase because it is made up of chitin. After doing this, after breaking the cell wall, we are getting that cell membrane. Cell membrane is destroyed. Cell membrane is destroyed by, second thing, cell membrane is destroyed by detergent. Cell membrane is broken down by detergent. So detergent is used to break the cell membrane because this detergent is having some chemicals, which are used to break the peptide bond in between protein and the lipid, what is the main component of cell membrane. We know that cell membrane is made up of lipid and protein, and this detergent is having some chemicals what is called sodium didosyl sulfate. Okay, so these things, another chemicals used to break the peptide bonds or proteins which are the main component of the cell membrane, and cell membrane is destroyed, along with the nuclear membrane is also destroyed. Sodium chloride is also used. Sodium chloride. This sodium chloride, which are non-iodized sodium chloride, is used because it helps to bind the phosphate group of the DNA, and after that ethyl-ethanol, after that chilled ethanol is used. After that chilled ethanol means cold ethanol is used. Okay, when cold ethanol is used, then we are getting that DNA. This DNA we are getting in this thread-like structures we are getting. Thread-like structure is obtained. So in this way we take out the DNA from this cell. So in this way we take out the DNA from the cell. We take out DNA from the cell. That is called isolation of DNA. So in a nutshell, we can tell you that what DNA which contain the desired genes is present in a plant cells or any microbes or any fungus, that has to be taken out. That is called isolation. And for that we have to break the cell wall by some enzyme, cellulase. After that cell membrane by detergent, because detergent is having some chemicals to destroy the peptide bond, and lipid and proteins are separated, and cell membrane and nuclear membrane are destroyed. After that what we are getting the thread-like structures. When we apply cold ethanol, then this cold ethanol, in this cold ethanol DNA is insoluble and we are getting thread-like structures. In this way the thread-like structures is obtained and isolation of DNA is complete. Next comes fragmentation of DNA by enzyme. Then what DNA is obtained? What DNA is obtained by the process of isolation that may have some protein with it. That protein is to be removed from the DNA to purify it, and for this purpose we have to use some enzyme called protease. Except protease we can use except protease enzyme we can use some extract of papaya, because papaya contain one enzyme called papain. This also dissolve protein, and in this way we can purify the DNA. After purification, I am going to the third part, that is fragmentation. That is fragmentation of DNA. That means what? This DNA is to be fragmented. This DNA, what is this part? This DNA is the structure of DNA. This DNA is divided into different segments. Because we have to make, the scientists have to make small segments which are carrying that property. That is that process called segmentation or fragmentation. By the process of, we have to make segments by the process of fragmentation. By the process of fragmentation, we have to make separate segments of this DNA, and which DNA is having that particular gene or particular desired gene, that has to be isolated. Next I am coming, for this process of fragmentation, one enzyme is required, that is called restriction enzyme. That is called restriction enzyme. This restriction enzyme belongs to a group of enzyme nuclease. This restriction enzyme mainly two types. One is restriction endonuclease and restriction exonuclease. Restriction exonuclease. So from this term itself, it is clear that it cuts This cuts the DNA at the middle portion, in the inner side, and exonuclease cuts the DNA in the end, at the different ends, either this end or this end. That is called exonuclease. So exonuclease and endonuclease are the part of restriction enzyme. This endonuclease cuts the DNA fragments at the middle or inner side. That's why it's called endonuclease. And exonuclease is that enzyme which cuts the DNA fragments on any side, either left side or right side. That's why it is called exonuclease. Out of these two types, there are a restriction endonuclease is very important. That is restriction endonuclease. Restriction endonuclease. So this restriction endonuclease has certain property to cut the DNA fragments. Number one, it cuts DNA into different or into many fragments, and it helps to make the sticky end. This sticky end helps to join with the vector's DNA firmly. This sticky end helps to join the plasmid DNA firmly. These restriction enzymes are of three types. Out of these three types, this restriction enzyme 2 is very useful for recombinant DNA technology. Type 2 of restriction endonuclease is very much helpful for recombinant DNA technology. So it is very important. And another thing is that this restriction enzyme, where this restriction enzyme cuts. So first restriction endonuclease is HINT2, which was isolated and discovered by these three scientists, Smith, Wilcox and Kelly, in the year 1962. There are different types of, more or less 900 restriction enzymes have been isolated so far. 900 restriction enzymes have been isolated. Now I am going to tell you that how to give name of this restriction enzyme. For an example, I am telling you, one of the restriction enzyme is EcoR1. So these names, this is the one of the restriction enzyme which has been isolated from one bacteria called Escherichia coli. So this restriction enzyme or endonuclease has been discovered from the one bacteria that is called Escherichia coli. So how this name has been developed? E for Escherichia means genus name. Genus name Escherichia. CO is the species name. Species name coli. Species name here coli. Understood? And R is the strain. R is the name of the strain. Here strain is RY13. Is RY13. That's why it is R. And 1 stands for, this one stands for the sequence of isolation. Sequence of isolation. In which sequence it has been isolated? Sequence of isolation. So from this name of EcoRI, how to give the name of this restriction enzyme, it will be clear to you. If you see the first letter is from Escherichia, E, the first letter of genus Escherichia. CO is the first two species, that is coli. R is the strain. Here strain is RY13, and 1 is the stands for sequence of isolation. In which sequence it has been isolated? That is the, in this way, the restriction enzyme name has been given by the scientists. Where this EcoRI cut, they will find the palindromic sequence. What is palindromic sequence? I am telling you. Suppose it is a DNA structure. Here 5 dash, here 3 dash, here 5 dash, here 3 dash. Here suppose guanine is there, after that adenine, after that thymine, after that cytosine. Okay, so these are the arrangement of the nitrogen base, and here also you see G, A, T, T, C. So you see where this found, the arrangement is same, G, A, T, T, C, and for the list G, A, T, T, C. So this, if you go left to right, what is the arrangement? If you come right to left, same arrangement. That is called palindromic sequence. Is it clear? So this is very important. Palindromic sequence is that arrangement. If we go from left to right, what arrangement is there? Nitrogen base is there. If we come from right to left, the same arrangement is there. That is called palindromic sequence. And this eco-RI will cut over here. This eco-RI, this enzyme will cut over here, here and here. Understood? So when if it is cut like this, then and if you cut like this by this restriction enzyme, so this restriction enzyme is called molecular scissor. Molecular scissor, because it cuts it. So if it cuts, then what type of structure we'll get? We'll get that first one is one strand will have like this, T, T, A. This is one strand. In another side here, one G will be there, this G, and upside will be C, T, T, A. You see, this is one portion is hanging out, one portion is hanging out or stretched. One end is stretched out, one end is stretched out. That is called sticky end. Try to understand this part. This is one portion and that is another side. Okay, here this portion is stretched out and here this portion is stretched out. That is called sticky end. This is called sticky end. This sticky end or formation of sticky end is the unique role of this restriction enzyme. If it is sticky end, then they will combine or join with the plasmid DNA family. This has to make the sticky end, and the unique property of this restriction enzyme or every restriction enzyme to cut the DNA and make sticky end. So it is clear that when the restriction enzyme cut the DNA, it makes two parts. One is like this, another is like this. So this is called sticky end. As I told you over here, here G is there, here this portion is still about C, T, T, A. And here also only G is present, here C, T, T, A is present. So when this restriction enzyme also cut the DNA plasmid, the plasmid of bacteria or vector. Okay, vectors plasmid, that will also form the sticky end. That also form the sticky end, like suppose like this. Okay, you see here one end is stretched out, here upper end is stretched out. So this will be fitted over here easily. Okay, so this will be fitted over here easily and firmly by the enzyme. Suppose this will come and mix over here like this. So this will be joined firmly. Here the ligase will join it firmly. Okay, and when this, it is the plasmid DNA, and it is desired DNA. And when this is having plasmid DNA, takes this one within it, it makes recombinant DNA. Okay, that in this way recombinant DNA will be formed. In the next video, I am going to discuss gel electrophoresis. So if you want to learn from it, please subscribe my channel and share with your friends. Thank you very much for being with us for a longer time.